Waves lap at a rocky shore. A woman gathers shellfish.
It's just very fulfilling to use science as a way to protect marine animals and ecosystems. Working with the Park Service, you get to go to these sites and see what you're working to protect.
A brown bear moves slowly across the rocks. The National Park Service logo appears. Beneath it appear the words, "Changing Tides, investigating the ties between intertidal invertebrates, brown bears, and people in Lake Clark and Katmai National Parks."
My name is Katrina Cunahan. I'm a scientist at the Alaska Sea Life Center.
On a rocky beach, Katrina gathers shellfish.
The goal of this project is to use bivalves, so razor clams, mussels, and a couple other clam species, to look at how the coastal ecosystem in Lake Clark and Katmai are changing.
Clouds move over an alpine lake in fast motion. On a beach, a brown bear digs in the sand.
This is important for humans and wildlife, so the bears depend on these bivalves as a food resource. And so if something happens to the bivalves, that could impact the bear population.
In shallow water, a shellfish sits in the sand. On the beach, Katrina digs for shellfish.
Bivalves are very useful because they live in that environment and they don't really move from the area that they're in.
She lays a shellfish in a pile in the sand.
They're filter feeders, so they filter out small particles in the water. That's why they're so useful, is their ability to filter feed allows them to basically concentrate what's in the water around them.
She rinses the shellfish in a bucket of water, then puts them in a mesh bag.
That also includes things like pollutants and bacteria and stuff like that.
Outside a log cabin, Katrina pries open a shellfish with a knife. In a lab, a row of specimens in test tubes.
For the lab work that the Alaska Sea Life Center is doing, we have several experiments that look at different aspects of the mussel's health. So the first thing we do when we get them back in the lab is we measure their width, their length, and their height, and their weight. These are what we call morphometric measurements.
In the lab, a technician cuts open a shellfish. On a scale, a shellfish sits in a petri dish.
We can do several calculations based on these measurements. One of the measurements we take is called condition factor, and this is basically how much tissue the bivalve has versus how big it is. And essentially gives us an idea of their nutritional status. Have they been getting enough food to grow? If they're not making enough tissue and they're not growing well, then that can indicate that there's a problem at the site.
A technician pulls apart a shellfish sample in a petri dish.
So after we take those, the morphometrics, then we dissect them. One of the experiments we're doing is called the P450 assay. We're concerned about oil exposure, so hydrocarbons from either vessels in the area or, worst case scenario, be an oil spill. And so P450 is an enzyme that processes hydrocarbons. And it can also process other man-made chemicals. We can look at the enzyme activity and see if they've been exposed to hydrocarbons.
With a syringe, a technician injects fluid into test tubes. In a boat, a woman with two men.
Every organism has DNA. It's the blueprint for the whole organism and how the organism functions on a daily basis.
On a beach, the woman digs in the sand with a shovel.
From the DNA, a template is made into messenger RNA. This is what we're measuring. The messenger RNA dictates which proteins will be made for which function.
She removes the shellfish from the sand.
My name is Liz Bowen, and I'm an ecologist with USGS. We're using a tool called gene transcription. Let's say we collect a razor clam, take it back to the boat, we open it up, and I take a tiny piece of gill tissue and a tiny piece of the mantle. And then we stick it in a special buffer that stabilizes it so nothing else changes, bring it back to the lab at UC Davis, and we do the little gene transcript magic on it and see how many transcripts for which gene are being produced.
In the lab, technicians cut open shellfish, remove tissue samples from them, and put the samples into test tubes.
The technique itself has been in existence for 15 to 20 years and used primarily in human medicine. So when you go to the doctor, they'll run these tests on you, they look in your blood and see what your transcripts are. It's being used a lot for gene therapies for cancer. So we've adapted it, modified it to use on the razor clam and the mussel. It can tell us whether the organism is sick. It can tell us whether there's not enough food. Are there pathogens, bacteria and viruses? Is there a toxin in the environment?
A technician examines a computer readout of the scanned tissue.
It's an incredible opportunity for all the scientists involved and the science community and the public to gain a lot of information about an amazing place and ecosystems that we rely on.
Snow-capped mountains rise up above an alpine lake. On a beach, a brown bear digs in the sand.
So many animals are connected with this that it's a great opportunity for all of us to learn more.
From the bank of a pond with a small waterfall in it, a woman and 2 children watch brown bears in the water.
Our ability to preserve these national parks is important for everyone who wants to come and enjoy them in the future.
We're seeing huge changes with global warming. How is that going to affect all of this out here?
Carrying her shovel, Liz makes her way along a beach.
And certainly, you know, we can't stop the global warming, but we can help mitigate what's going on. Things are starting to change now. Let's do something about it.
The National Park Service logo appears. Credits: described video by Descriptive Video Works. Direction: Jim Pfeifenberger. Videography: Jim Pfeifenberger. Katie Kritz, Annie Langen, and Xochitl Rojas Roca. Special thanks to Katrina Cunahan and Liz Bowen.